Hello, my name is Celine Lajoie and I'm a PhD student at McMaster University in Dr. Karen Kidd's lab and a Wildlife Conservation Society W. Weston Fellow. I am studying the effects of forestry on mercury accumulation in stream food webs in northwestern Ontario. Forest harvesting activities such as logging and road construction can impact surrounding aquatic systems. Streams within forests are ecologically connected to their terrestrial landscapes. For example, aquatic invertebrates or insects that live within streams often rely on leaves from trees in the forest as a primary food source. Also, forests can play important roles in maintaining water quality. Forest harvesting practices can actually disrupt these important services. Studies have shown that mercury, a contaminant stored in forest soils, can be mobilized by harvesting activities and transported into nearby streams. Mercury is readily converted to its more toxic form, methylmercury, and accumulates in aquatic organisms. This can lead to dangerously high levels in fish and pose health risks to the humans who eat them. Harvesting companies have methods in place to reduce environmental impacts called best management practices, or BMPs. These include leaving areas near the stream unharvested and harvesting in the winter, for example. While these do reduce environmental impacts, we don't know how they affect mercury accumulation in stream food webs. This project aims to fill this gap in the knowledge by figuring out how forestry impacts stream food web structure and function, and also by determining the effects of forestry on mercury bioaccumulation in boreal stream food webs and how effective best management practices are in mitigating its risks. This research takes place in the Kenora-Dryden region of northwestern Ontario, which is about 3 hours northwest of Thunder Bay. We are collecting samples from 12 streams located across the Centrefire, de Norwick, Snowfall, and Dessert regions. We are collecting samples over a time period of 3 years, so from 2019 to 2021. Samples are collected twice in the summer and once in the fall, although sometimes the fall can feel a bit like winter. Samples will be collected pre- and post-harvest, and this will allow us to make comparisons in food web structure and mercury levels pre- and post-impact. From each stream, we collected food sources that consumers would be eating. We collected fine particulate organic matter, we collected leaves, and we collected biofilm. My favorite part of the project involves sampling macroinvertebrates in streams with kick and sweeps. We then grab our tweezers and sort the bugs on site. Here we can see a couple of bugs wiggling around inside ice cube trays. One of the reasons I really enjoy this part of the project is because we get to see a lot of diversity that the boreal streams hold. Plus, they're pretty cute. We then bring the bugs back with us to the lab to identify them to genus using dissection scopes. Another part of sampling that I really enjoy is fishing. So here's a video of us catching fish, and you can see in this video that I'm wearing a very large backpack. That backpack does not make me a Ghostbuster. That's actually a backpack electrofishing unit, which is allowing us to shock the water and stun the fish. This makes the fish easier to catch. We're then able to identify the fish on the stream bank, and we bring the fish back along with all the other samples that we've collected to the lab to do mercury and stable isotope analyses. Sometimes fishing doesn't always go as planned and you need to improvise. So one day, after an unsuccessful fishing trip, we decided to set up a bunch of minnow traps. When we came back the next day, we were really excited to see what we had caught, which turned out just to be a bunch of frogs. What excites me most about my research is how it is part of a huge collaborative effort between universities, government agencies, and forestry companies. We are taking a multidisciplinary approach looking at mercury dynamics in forested watersheds in Canada's boreal. I love my project, and it is a piece of a larger puzzle that will help us understand how mercury is affected by forestry and which best management practices may mitigate mercury risk to aquatic ecosystems. I am very excited by the conservation potential of this project as it could lead to the development of more sustainable forestry guidelines and policies. I also love the fact that I get to work in such a beautiful area on such an interesting question. I am very grateful to the Wildlife Conservation Society Canada and the W. Weston Foundation for your generous support. Your generosity was instrumental to our fieldwork this year and even helped us look at the project from a new perspective. Thank you again, and I am very excited to share our future results with you.